Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast. And the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. While great progress has been made in understanding the human genome, its functional counterpart, the human proteome, remains relatively unexplored. In part, that's because advances in the tools to identify the far bigger universe of protein variants has been lacking. Seer Bio has developed a platform for large-scale proteomics studies that it says is both unbiased and scalable. We spoke to Omid Farokhzad, Chair and CEO of Seer Bio, about the state of proteomics, how Seer's technology works, and why it can help advance our understanding of health and disease. Omid, thanks for joining us.
Thank you so much for having me.
We're going to talk about Seer, the challenge of proteomics today, and how Seer's technology is being used to understand the proteome. Before we talk about Seer's technology, though, I thought we could take a step back and talk about where we are in understanding proteomics today. As you think about the work on the Human Genome Project and the progress we've made there today, where are we in terms of understanding the human proteome?
Then if we look at the genome, it took us 15 years and just shy of 3 billion to sequence the first genome. And then with the advent of next generation sequencing, cost dropped to 200,000, and now we're at 1,000 with visibility to $100 genome there. And frankly, $10 genome may also not be too distant in the future. The consequence of all of that is that we've sequenced over 1 million genome, over 10 million exomes, and in aggregate, at the population level, we've now identified over 695 million genetic variants. And the thing is that while we've identified these variants at the genomic level, we know a tiny bit about what these variants mean in terms of their biological function. And how does that genotypic changes actually affect function at the population level within the proteins? And remember, our genes are largely static, and they're an excellent indicator of our risk, but proteins are dynamic and a true indicator of our status. And we just have very, very little information at the protein level. And frankly, the reason for it is that proteins are complex, and I'm sure we're going to get into it, and we just haven't had the tools to get into the depth, the breadth of the complexity of the proteome, and that lack of information hasn't really enabled us to connect the genotype to phenotype.
Well, what's the reason for that? Why have we had such a challenge connecting genotype to phenotype and understanding the function?
Well, look, That's because, in the simplest term, biology is complex. I mean, we're all born with about 20,000 genes. All the cells in our bodies, every cell has the same genes. But as you go from genes to RNA, and then from RNA to proteins, every step adds complexity. So, for example, you go from about 20,000 genes, but because those genes can produce many different RNA transcripts, our transcriptome could be as large as 200,000. And then by the time you go from these transcripts, and some of them actually won't code for protein, but many do, you end up with a lot of proteins, and then proteins themselves get modified after synthesis, post-translational modification, if you would. And the complexity of the proteome could be million plus different proteoform or protein variants that are coded by the same 20,000 genes. So as you go from the 20,000 genes toward the protein, you end up with a million different variants of protein. And by the way, that estimate varies depending on whose papers you're reading, but certainly complexity that is much, much larger than at the genomic level. Now, these protein variants, proteins come together and form complexes, and these complex of proteins exert a biological function, and different variants of proteins can actually come together in a distinct and unique way during health and disease. And so if you imagine the complexity that we're talking about, the tools that we've had at our disposal to look at the proteome just haven't allowed us to get to this depth and breadth of the complexity of the proteome to date.
So we've seen these remarkable advances in the genomic tools. We've shattered Moore's law with those. Where are we in our advancing of tools to understand the proteome? How well suited are they to the task at hand?
Look, I mean, the approaches folks have taken to the study of proteins at the broadest level, I would say, fall into two buckets. You can approach proteins in a targeted way or in a, quote, biased way, or you can approach proteins in an untargeted or unbiased way. So a targeted means that I know what I'm looking for, and I'm going to create an analyte-specific reagent or an ASR, and to go and interrogate that protein. Now, any ligand can be an ASR. So a classic one would be an antibody, but virtually any ligand class, so nucleic acid ligands like aptamers or many different protein scaffold ligands like nanobodies, affibodies, adnectins, demantis, those are all different classes of ligands. You can select a ligand for a protein of interest, and then you can go and repeatedly interrogate it. So that's a biased approach to proteomic. Now, if you look at it from an antibody perspective, these biased approaches can be, again, divided into two buckets. They can be a monoclonal approach, or they can be a polyclonal approach. In a monoclonal approach, I have an ASR that always binds to a very specific part of a protein, and I can go and interrogate that. And by virtue of the fact that that ASR is binding to a very specific part of a protein, it could actually have utility in clinical application. And in fact, many of these monoclonal targeted approaches are perfectly suitable for a single protein or low plex protein clinical utility. And in fact, many of the proteomic diagnostics today will take an approach like that. You can also have an ASR approach, which is a polyclonal, if you would, approach, where you would have many different ligands against that protein. Now, the challenge with the monoclonal approaches are they're excellent because you can actually identify a specific protein variant, but they're very difficult to make. So a typical monoclonal antibody would take months to make. Now, a polyclonal approaches to a targeted proteomic approach are much more scalable, You can actually develop a polyclonal ligand to a protein very quickly. You can even do it in parallel and do multiple at once. But the challenge is that you are not able to distinguish different variants of a protein when you approach it in a polyclonal way, number one. And number two, they don't have applications, at least that I'm aware of, in terms of clinical utility. The targeted proteomic approaches that are used in clinical applications are monoclonal applications, where repeatedly you can have the same exact protein from the same hybridoma, if you would, making the same antibody, and then you can go interrogate it. With polyclonal approaches, every time you kind of make it, there is batch-to-batch variability because those polyclonal products that you produce will be different from each other, and they don't have utility in clinical application. So those are targeted approaches. Now, a polyclonal targeted approach can actually have utility, for example, in translational research, but at least to date, it has not had traction in clinical application. Or you can approach proteomics in an unbiased way or a non-targeted way. And in that context, you're saying that I don't know what is important. I want to look at as much as I can find the totality of the information, if you would. And in fact, that is how new biological insight is gained. Now, the non-targeted approaches or the unbiased approaches to proteomics have been around for a long time, typically involve a liquid chromatography and a mass spec. And the challenge that they've had is that they just don't scale. So you could look at the totality of the information, if you would, but because you can't do it at scale, it's unlikely that you're going to be able to gain the types of biological insight that we were able to gain when we looked at genomics at scale. Now remember, before we got unbiased genomic at scale, and by that I mean next generation sequencing, I can do large number of samples, I can do them at the cost and in a time frame that makes it possible to do large scale population level studies. Before we had that, we still had targeted genomic approaches. That's basically the PCR. You could PCR amplify a fragment, and then you could have used, the Sanger didioxane method of sequencing it. By the way, I've done my fair share of pouring those gels and running those sequencing gels myself early in my career. Now, the challenge was that was difficult to do and it didn't scale. When unbiased genomic approaches came, the utility of targeted genomic approaches did not go away. In fact, as we were able to gain new biological insight by the magnitude of the information that was becoming available to us, the utility and the application for targeted genomic approaches like a PCR actually increased because we now had more interesting things to go and interrogate in a rapid way using targeted approaches. So when I look at proteomics, where the real biological insight, where that massive part of the unmet need, the total addressable market, if you would, in the proteomic exists, is getting all of that content, cataloging all of that content, which is actually going to create end markets that probably don't even exist today. But as you do that, there will be other applications, and a subset of those will be used in a targeted way. And in fact, I think the utility for targeted proteomic approaches will actually increase, as unbiased proteomic approaches allow us to get deeper and deeper into the proteome and get to deciphering, if you would, the complexity of the proteome, which to date has remained largely untouched.
Want to take a slightly deeper dive into that terminology of a biased approach. The traditional way that researchers have studied the proteome is through the use of analyte-specific reagents, or ASRs, and this is called a biased approach because it's targeted. Is the suggestion that they could only find what they were looking for?
That is exactly right. Look, if I take an antibody and you have to imagine with me, uh, an average human protein is 472 amino acids long. An average ASR binds to an epitope that is only 5 to 8 amino acid long. So if I make a monoclonal antibody against a given protein, that means that for that, on the average 472 amino acid long protein, I am binding to a tiny portion of that protein, which is the 5 to 8 amino acid that is my binding site. Now, that protein could change anywhere else other than the binding sites during health and disease. It could be post-translationally modified. There could be genetic variants that changes amino acids anywhere else in that protein. I would never be able to see that in terms of the variance of that protein that exists using that approach. Now, monoclonal approaches can be excellent if you know exactly what you're looking for. So for example, let's say I have a kinase, and this particular kinase gets phosphorylated at a particular serine residue in that protein. I can develop a monoclonal antibody that will bind to the phosphorylated version of that protein at that serine residue, but not to the non-phosphorylated form of the protein at that same residue, right? So I can have the monoclonal antibody. So monoclonal antibodies are excellent if you know what you're looking for. Now, polyclonal approaches, unlike monoclonal approaches, actually are incapable, fundamentally not possible, to look at variants of the protein. Why? Because as you generate those reagents, you're making many different ligands against that 472 amino acid long protein. And any one of the various different polyclonal antibodies can bind to that protein. So as that protein creates variants of it, whether those variants are at the level of amino acid changes or whether those variants are at the post-translational modification changes, perhaps one of those antibodies may not bind, but other in that pool of polyclonal does bind. And so the net is a positive binding, incapable of differentiating the different variants of the proteins together. So the limitation of targeted approaches are the following. One, in the case of monoclonal, they don't scale because it's very difficult and time consuming to develop monoclonal for each of the variants that exist in the proteome. When you look at it from a polyclonal approach, which does scale, you are limited in the fact that you cannot identify variants of a protein. So in essence, if the genome was such that one gene codes for only one protein, and therefore the totality of our 20,000 genome would encode 20,000 protein, then a polyclonal approach would have been excellent because A, it is scalable, relatively speaking to the monoclonal approaches, and B, I could pick that up. But the fact that we go from 20,000 genes to million plus protein, the inability to identify variants of those proteins is a significant handicap when it comes to discovery and gaining new biological insight.
There have been so-called unbiased tools researchers can use, but you say these are limited because they're not scalable. Can you explain?
Yes, of course. If you look at the existing unbiased approaches to proteomic, typically these involve a liquid chromatography and mass spec. And the reason for those tools being used is that not only is the proteome complex in terms of the structure of the proteins, the proteome is also complex in terms of the quantities, if you would, of various different proteins in a biological sample or a biological matrix. So for example, if you take the plasma, within the human plasma, from the most abundant protein to the least abundant protein, there is over 10 order of magnitude in concentration difference. So albumin is the most abundant protein in plasma. Albumin alone represents by mass 50% of the total protein in plasma. Now, the next 20 or so abundant proteins after albumin in plasma, together with albumin, represent by mass over 99% of the total protein in plasma. And what that means is the balance of the plasma proteins, which is thousands of proteins, is that 1% of protein by mass. And so what scientists and researchers have done in order, in an unbiased and untargeted way, to interrogate the proteins, what they do is they create these separation techniques, like, for example, immunodepletion of the abundant proteins, fractionations of many of the subsequent proteins in the sample. And then each of those, if you would, fractions that gets created is then put into a mass spec for analysis. And the problem is that the amount of time it takes to do these workflows, it limits the size of the study a researcher can run. And so invariably, the researcher has to, without an exception, make a choice of how deep do they want to go into the proteome, and then, versus the size of the study they can do. And that compromise is unfortunate, because if you want to gain insight in biology, you have to go deep and you have to do it on a large number of samples. The reason we've identified over 695 million genetic variants is because we have now sequenced over 1 million genomes. We have sequenced over 10 million exomes. The very first one that we did informed us very little. It's all the subsequent one that added to the enormous amount of information that got gained. So we have to be able to do unbiased proteomics really at the same scale and speed as we can do genomic studies in an unbiased way. By the way, when you do that, something awesome happens. And that is that you now impedance match the generation of unbiased genomic with unbiased proteomic. And when you do that, you really enable the field of proteogenomics, which really enables you to go from genotype to phenotype to really make sense of the biological content that are being generated, both in terms of genomic side and proteomic side, really in a, if you would, a self-fulfilling cycle where proteomic information adds value to genomic content, and then more genomic information drives more demand for proteomic information. And so to really get there, you gotta impedance match our ability to access the genome in an unbiased way at scale with the proteome in an unbiased way at scale. And I think we are basically at that watershed moment in time where that is now becoming possible, exactly as it became possible 15 years ago in the genomic world.
Seer is using nanoparticles with a magnetic core. What is a nanoparticle?
Nanoparticles are basically spheres, if you would, very, very small spheres, small enough that you could put about 80,000 of them side by side in the cross section of your hair. And by the way, I spent 20 years of my life playing with these nanoparticles for a range of applications in medicine, developing targeted drugs delivery system, developing vaccine, and then in the context of Seer, for protein sampling. What is interesting in the way these nanoparticles behave is that, frankly, they kind of follow nature. By the way, many of the awesome innovations that happen in science and medicine is us in some way, shape, or form mimicking what nature has taught us. If you look at the way proteins interact with themselves, remember early on I said that proteins function by coming together and forming complexes, and as a complex, that machinery is able to function and drive a particular application that collection of proteins were designed to do. Now, how do the proteins come together? Well, they come together because the physical chemical properties of one protein matches the other protein. And by that I mean like the carbon, nitrogen, oxygen molecule of one is oriented in a way that it kind of fits into the other one. So that physical chemical relationship bring them together, but also in nature, two other factors plays part. One is that temporally, those two proteins are expressed at the same time. And then spatially, they're also expressed near each other. So it could be in the same cellular compartment, it could be in the same tissue, same organ, but the proximity of those proteins together being expressed at the same time and physical-chemically fitting each other makes them come together. Now, that happens in evolution over millions of years. You can actually accelerate that process with data science and machine learning in a lab where you can design nanoparticles with specific physical chemical surfaces that are distinct from each other. So a nanoparticle, depending on its physical chemical structure, will sample proteins in a complex matrix of proteins in a very unique way that is a signature of that nanoparticle. And so what that means is, if I take, for example, a biological sample, and let's say I take your blood, and I expose your blood to a particular nanoparticle that's got a very defined physical chemical properties, I will sample a set of proteins in an unbiased way. I will do that robustly and reproducibly. Different operators, different hands, different instruments, different labs. I'll get a very similar type of sampling. So basically, Daniel, mimicking what nature has done in terms of the way proteins interact with each other, you can basically recapitulate that exact thing in a laboratory by the way you engineer these nanoparticles. So Daniel, if you think of the way proteins bind to each other, they bind to each other because the physical chemical properties of one protein matches the physical chemical properties of another protein, meaning the way the carbon, hydrogen, oxygen atom of one protein is lined up such that it actually matches, the atoms of the other one in a way that it kind of fits into a pocket with each other. Now, in the case of proteins binding with each other, two other elements are at play. One is that they're expressed at the same time. So there's a temporal component in the way those proteins see each other. There's also spatial expression in that those proteins are expressed not only at the same time, but in the same tissue or in the same cell or cellular compartment. Now, that's basically biology and evolution to bring those proteins to come together and exert a biological function. You can learn a lot from biology, and you can essentially mimic those same kinds of bindings, if you would, in a lab using nanoparticle technologies. So you can design these nanoparticles with distinct physical chemical properties. If you imagine the universe or the canvas of physical chemical design is almost infinite in terms of what we can do, and we can design these nanoparticles with unique physical chemical structures and then explore what is it that nanoparticle binds to and picks up in a highly robust and reproducible way. Now, that binding is actually quite robust. So if I take the same nanoparticle and a same biological sample, let's say your plasma, for example, and I do the assay in different days by different operators in different hands using different instruments, I will end up with a very similar pattern of protein sampling. So now I take your blood again, your plasma, and I now change the physical chemical properties of a nanoparticle. I basically design a different nanoparticle, and I come back and I sample your protein again. And now I have a different pattern of protein sampling than the first nanoparticle. As you can imagine, I'm not doing any depletion and fractionation. This is basically a binding assay that's happening. And by just adding different nanoparticles, I can basically sample the proteome across its entire dynamic range in a highly robust and reproducible way. Now, the way the nanoparticles bind proteins is actually quite interesting in that sampling isn't as a function of what is the most abundant protein. If it was, then the nanoparticles would largely sample albumin and other abundant proteins. But the binding to the surface of the nanoparticle is driven by not only the abundance of that protein, but also the affinity of those proteins for the surface of the nanoparticles, such that maybe a low abundant protein that has a higher affinity for that nanoparticle surface, it would actually occupy space, displacing a higher abundant protein with a lower affinity, and then that nanoparticle samples it. So if you look at the sampling that our nanoparticle does in a biological sample, we can essentially sample across the entire dynamic range of the proteome, seeing everything, of course, from the most abundant protein like albumin, but seeing very low abundant proteins, like cytokines. And again, that sampling is robust, reproducible. If you look at the coefficient of variance of these assays, quite tight. And let me know if that answers the question, Daniel, and I'm happy to expand as helpful.
How good are these at finding proteins that may be in low concentrations?
Well, look, you just, as a scientist, you just have to follow the data, right? So if I look at the way a panel of our nanoparticles sample the protein, and I compare that with existing approaches, depletion fractionation approaches, to go into deep unbiased proteomic, we can go deeper into the proteome, and we can do that with a better reproducibility in terms of our assay and CV. So we can get low abundant proteins, Daniel, going down to the proteome, proteins like cytokines, as well as sampling other proteins that are relatively more abundant.
This is part of a suite of products. How do you move from sample to result with your platform?
So if you look at what the product suite of Seer is, it's the Proteograph product suite. It comprises our consumables that includes our proprietary nanoparticles, but there's other reagents in that kit. And then there is an automation instrument. This is a fluid handling instrument. It's an OEM instrument designed for us. It only runs our assay. And then there is a software suite that helps the scientists go from biological data, if you would, to biological insight. Now, there is a missing link here, which is the detector. Our detector of choice is a mass spec. We are actually detector agnostic. And by that, I mean the Proteograph instrument sits upstream to a mass spec, and it works with any different types of mass spec. In fact, in our own labs at Seer, we have thermal mass specs, Bruker mass specs, SciEx mass specs, and no reason that other mass specs won't work. And in fact, the Proteograph product suite is actually mass spec, not only mass spec agnostic, but it's detector agnostic in totality. And by that, I mean, if there is another detector some point in the future that could be developed that can look at the proteins in an unbiased way, then the workflow of the Proteograph, the terminal workflow of the Proteograph could be tweaked to then work with that detector. So what happens is you have the Proteograph, the consumable is basically a razor blade model. A customer buys the instrument, and then they use the reagents. The instrument is a 7-hour workflow, of which there's 30 minutes of hands-on time for the operator to load the machine. Then is 6.5 hours of fully automated time where the machine runs the assay. You process 16 samples in parallel, and then those samples come out of the Proteograph and they go into a detector. Again, I said we're detector agnostic, but currently the detector of choice is mass spec. The reason for it is that there is 15,000 mass spec installed globally that does proteomic studies and proteomic work. And so there's a large number of installed base that the Proteograph can sit upstream to. And then as the information comes off the mass spec, it goes into our own data suite for analysis that the customer can use in understanding basically the content that they're getting out of the detector.
And how scalable is this approach? What can researchers do with Seer's technology that they can't do with other approaches?
I said early on that essentially what Seer has made possible is to impedance match or access to unbiased genomic content and unbiased proteomic content. So you can actually, one Proteograph and 2 mass specs can roughly match the throughput of 1 NovaSeq instrument in about basically in the same time frame. So in about a 2-day time frame, 1 Proteograph and 2 mass specs can process about 48 or so samples, and the same throughput is a NovaSeq instrument that would do about 48 samples in terms of genomic content. So the scalability of it now matches basically the scalability that we have in accessing genomic content. Now, this is not an incremental improvement. So prior to Seer, the largest plasma deep proteomic studies, and deep is defined as a study that detected at least 600 proteins. The largest plasma deep unbiased proteomic study that we are aware of was a study with 48 samples. Now contrast that to the size of the genomic studies that we're doing. By the way, Seer actually published a paper in Nature Communication just about a year ago, last July. And in that paper, there was 141 subjects in that paper that the depth of the protein was to about 2,000 protein per subject. And the data acquisition part of that, is under 2 weeks, basically, to get to that level of data. So it now becomes possible, Daniel, to actually do really large samples, population level studies like we're doing in the genomics, as we move forward. As you know that the Proteograph product suite is now commercialized, and we're in the second of third phase of our commercialization. The first phase was the collaboration phase, followed by the limited release, which is the phase that we're in now, followed by broad release, which happens at the end of the year or beginning of next year. And if you look at our early customers, Oregon Health was our first customer. It was a collaboration customer. They received their instrument the end of last year. We've announced on our last earnings call that Oregon has now completed their pilot studies, and Oregon is now planning studies in the size of 500 to 1,000 sample study, which is a log or log and a half bigger than any prior study in unbiased proteomic that they could do in deep unbiased proteomic. So the scalability in terms of access to proteomic content that becomes possible using the Seer Proteograph product suite is radically significant advance in what was possible before.
As you think about driving commercial adoption here, what do you see as the biggest challenges?
Look, we're obviously on the very, very early part of this journey. I said we're at a watershed moment, the likes of which we saw in the genomic 15 years ago. Now, I wouldn't say it's going to take us 15 years in the proteomic space to get to where genomic is today, because many of our colleagues in the genomic space have paved the road for us that we're traveling on. So our slope of adoption is going to be sharper than what happened with genomic. Many of these customers already exist that are doing large-scale genomic content, that they're just looking for annotating those genomic information with proteomic information. And so they will readily adopt large-scale proteomic studies. But I would say, Daniel, we're at the early part of this journey, and execution is never easy. Now, I am so proud of the team that we have built in all different functions of this organization, a team that has been just on top of the food chain in terms of this life science and tools business, including my colleague, Omid Farokhzad, who's our president and chief operating officer. And Omid was previously at Illumina for 15 years, and before that at Solexa, which was acquired by Illumina. So much of the brilliance of the marketing that and strategy that Illumina did in terms of penetrating the market, eventually gaining 80% market share in sequencing, having launched 11 sequencers in the last 15 years. A lot of that was done by Omid, and I'm just thrilled to have an individual of that caliber and that level of insight leading our operation and leading our product development and leading our commercialization at Seer. This is launching these products and building these companies, frankly, is a team sport. And I'm super, super proud of the team that we've assembled. And by the way, a growing team at Seer.
Omid Farokhzad, Chair and CEO of Seer Bio. Omid, thanks so much for your time today.
Thank you so much for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.